Good afternoon, everyone, and thank you for tuning in to our webinar today. My name is Brian, and for those of you who don't know about us, we are a government-owned organization in Singapore that invests in and helps to build deep tech startups, talent, and communities. At SGInnovate, our work involves connecting with the global deep tech ecosystem, working with entrepreneurial scientists to bring their innovative research from lab to market, as well as developing deep tech talent. Today's event, Tackling Chronic Diseases with Cell and Gene Therapies, is presented by SGInnovate. Today, we'll be having a panel of amazing experts in this field who will be sharing their insights on the significance of using living drugs as a therapeutic, the key considerations in its application and manufacturing, patient and public perspectives on cell and gene therapy, and the market opportunities and challenges. I also like to encourage our audience here today to share their thoughts on this topic and engage with our speakers by using the Q&A box below. And now, with no further ado, I'll now pass the time over to Mayura, who is the Director of Investor Relations and Communications at Arsenal Pharmaceuticals. Mayura, please.
Thank you, Brian, and thank you for the opportunity to discuss this very exciting topic today in our panel. As you mentioned, I'm currently at Aslan Pharmaceuticals. My current role is focused on communicating the development of our assets to both in inflammatory diseases, to both investor and medical communities. But prior to Aslan, I was actually involved in spinning out a technology in the cell and gene therapy space in Singapore. So this topic is quite close to my heart, and I think it's a very exciting space. And I'm very happy to have great panelists today with us to talk about their experiences and some of the challenges in this area. So maybe I'll just give a quick overview of the topic today. It's a vast space. The cell and gene therapy field is growing rapidly. And bringing truly revolutionary innovations to patients. We have seen a number of growing therapies approved for oncology as well as rare diseases, and there is vast potential in extending these applications to chronic diseases and conditions. But of course the path is not so straightforward. So that's what we'll be discussing in our panel today. So let me introduce our panel. We have Dr. Li Ong, Regulatory Consultant at Health Sciences Authority. We have Dr. Anthony Tan, a Senior Research Fellow at Duke NUS, and Dr. Natasha Ng, Senior Research Fellow at A-Star and co-founder of Beta Life. So welcome to all our panelists, and maybe I can turn over to you to do some quick introductions of yourself and what your role is.
Sure. Hi, good afternoon. I'm Lily, Regulatory Consultant from Health Sciences Authority. So first of all, I would like to thank the organizing committee from SGInnovate for inviting me to this event to share the regulators' perspective on cells and gene therapies. So the branch that I'm from, the Advanced Therapy Product branch, we are responsible for product registration for cell and gene therapy products for supply in Singapore. Yeah, so I'm the quality reviewer for products for registration, as well as any changes that you may make to the product after the product is approved. So we also review those applications. So we have implemented the regulation on cell tissue and gene therapy products last March, which is like 18 months ago. So we start to receive application for product registration. So apart from product registration, I also actively involved in reviewing clinical trial application pertaining to cell and gene therapy products, which actually usually you will submit this application to our clinical branch. Yeah, so I will help them in the evaluation as well. So this is primarily my scope of day-to-day work. Yeah, thank you.
So maybe I'll go first. So I'm Anthony Tan. Good afternoon, everyone, and taking time to listen to us, basically. So I'm not from the regulatory side, I'm more for the scientific side. So I'm a Senior Research Fellow at Duke NUS, working on the study of hepatitis B immunology. And we have also developed TCR T-cell therapy against hepatitis B, and at the same time for hepatitis B related liver cancer. So a couple of, I think 6-7 years ago, we started a company, Lion TCR, in collaboration with our partners. And I'm now also serving as a scientific consultant of Lion TCR. And clearly, we have done applications for clinical trials in Singapore. So I'm happy to now have the opportunity to discuss the challenges we have, or maybe I should call it our wish list as a company of what could be done, what could be made easier, and so on and so forth. So I'm happy to have an active conversation.
Okay, hi everyone. I'm Natasha. Thanks to SGInnovate as well for having me. So I am working full-time in A*STAR in the Institute of Molecular and Cell Biology. I work in the Stem Cells and Diabetes Lab. So as the name suggests, I have been working in diabetes research for a very long time, since my PhD days. When I came to A*STAR after my PhD, I'm working in a stem cell lab, and that's where I started deriving pancreatic cells and pancreatic beta cells from induced pluripotent stem cells, or iPSCs. So that's where the platform with which we have done a lot of research studies, where we use these cells for disease modeling and discovery of cell biology. But in recent times, we have engaged in a few more translational projects where we try to develop these cells for cell therapy in diabetes. And together with my PI in the last couple of years, we have spun out a company as well. So this is an A*STAR spinoff. The name is Beta Life. As you can see, my affiliation. So I'm working for Beta Life as a scientific advisor and consultant as well. I spent a lot of my time doing the scientific development and doing the technical work, but on the side, I try to collaborate with Beta Life, the company, to develop these technologies in light of commercial strategies and regulatory strategies as well, to hopefully push this out. So I hope that what I can contribute to this discussion is probably mostly coming from the perspective of iPSC-based cell therapies, and of course, tackling chronic disease, which in our case is diabetes.
Okay, great. Thank you. Thanks to all the panelists. So, we've already heard some exciting therapies such as TCRT and iPSCs, and we'll dive into those in a minute, as well as, the regulatory requirements for clinical and registration stage. So the way it's a very vast topic. So the way we will tackle this is to first start off with understanding what are the types of these cell and gene therapy, how are they different to traditional drugs? So do a bit of a discussion on that before moving to really the crux of challenges that are faced in this space. So maybe, with that, Natasha or Anthony, one of you can start. I mean, you alluded to, the type of therapies you're working on and the diseases and the applications. But maybe you can provide a bit of information on how this therapy would work and what sort of promise or hope it would provide to the patients that current drugs maybe don't offer.
Okay, I'm happy to go first. So what we do in our lab in the company Beta Life is that we're trying to develop allogeneic cell therapy for treating diabetes. So there are a few subtypes of diabetes, and I think the first target sort of subtype of diabetes would be type 1 diabetes, which is well, in layman terms, is the more severe form of diabetes, where patients are diagnosed much earlier. It's an autoimmune disease, and essentially the pancreatic beta cells, which are the cells that secrete insulin, are being destroyed. And therefore, patients need to undergo lifelong insulin treatment. And this insulin treatment is basically, insulin injections daily, and having to continuously monitor your glucose levels. So this is, the current treatment, which I've alluded to, insulin injections. This is obviously not a curative treatment, and nobody likes to do that. And so the patient compliance is poor. And I think the challenge with being unable to control your blood glucose well is that you get all kinds of complications, microvascular and macrovascular complications. And this can eventually become cause a huge poor quality of life for the patients. And I think on the flip side, when you inject yourself with insulin, and if you don't fine-tune the amount of insulin you take in, that's where you get hypoglycemia, which is a dangerous dip in your glucose levels, and that in itself is a medical emergency. So I think what our company is trying to do, as well as a couple of others globally, is to develop iPSC-based cell therapy. So the iPSC technology has been around for more than a decade now, and it has obviously been used more in research studies, but in recent years, more and more research labs and companies are developing it as a source material for cell therapy. So what we do in the lab is to take the iPSCs and treat it with different kinds of media formulations to activate and inhibit certain pathways, to push them down, for our case, the endodermal lineage and the endocrine lineage, and eventually to derive beta cells that can express and secrete insulin in response to glucose levels. So this protocol in the lab takes about 1 to 2 months. So it is not trivial in terms of the cell manufacturing. It is very resource intensive in terms of the media that you need, the growth factors, the small molecules, and the compounds you need to turn them into a mature beta cell. But the hope is that with these beta cells, you can have an off-the-shelf cell therapy product. And the ways to administer it into patients would be to either infuse them directly into patients. So this will be a less invasive method. And the other method that has been currently used in clinical trials is also to embed them subcutaneously in an encapsulation device under the skin to help regulate sense your glucose levels, secrete the amount of insulin that's needed, and to basically cure diabetes. So I think I'm just trying to summarize I guess in a nutshell, how this whole sort of technical pipeline would work, for such a therapy.
Great, that's sounds very exciting to make pancreatic beta cells as an off-the-shelf. Very exciting. What about Anthony? Maybe you can talk a bit about TCRTs you're developing.
I think the strategy that of the TCRT is almost on the polar opposite of what Natasha is describing. So on one hand, she is trying to create a allogeneic off-the-shelf product with the idea to reconstitute cells that are not there, or at least dysfunctional. So the TCRT approach, on the other hand, it's the opposite. It's extremely personalized, it's autologous, and you are trying to create an army of cells to destroy things that you do not want in the body. So the typical example, which I think is extremely famous around the world, is the CD19 CAR for B-cell leukemia or B-cell lymphoma. So what is B-cell leukemia? B-cell lymphoma, essentially just B cells, uncontrolled growth of B cells, it's a tumor, it's a cancer. You want to remove them. So what you do is you develop T cells, put it into the patients where they will kill every single B cell in the individual. Well, the idea works from B cells simply because you're not going to die if you don't have B cells. That's it. But when you start to enter the realm of solid tumors and so on, then you start to deal with organs and things like that. Then you start to see the effects that you cannot just wipe out things. So what we are developing is essentially a treatment for chronic hepatitis B. So similar to a tumor, during chronic hepatitis B infection, instead of mounting a good immune response where you can destroy the infected cells, you don't. The cells are either recognizing the infected HBV infected cells and not doing anything, or these cells are completely gone in the individual. So the concept is, let's take other T cells from the individual and we make them recognize hepatitis B infected cells outside the body, and we put it back in with the hope that these cells will then start to kill the HBV infected cells. But the problem is we cannot really start with people that are chronically infected with hepatitis B, because like I said, now we are not dealing with B cells that you can eliminate and the individual is completely fine. Now we're dealing with the liver, which hepatitis B virus infects. We cannot just blast the liver and that's it. No, that's not possible at all. So since chronic hepatitis B patients, they live a good life for 20 years with no devastating consequences upon infection, we have to start the therapy in people that I would say, let's say, have a more severe condition, which is the outcome of long-term uncontrolled HPV infection, which is liver cancer or HPV-related liver cancer. The outcome of these patients, depending, is a spectrum, but once you reach a certain intermediate to advanced stage, the options for therapy are very minimal. You pretty much have transplant. Then, of course, you have more small molecules, new immunotherapeutic drugs that come up that have a myriad of effectiveness and efficacy. So that's kind of like the impetus for us to say, okay, let's develop a T-cell therapy. We want to develop it for chronic hepatitis B, but let's try it on cancer patients who are in more desperate need for it. And we have done phase 1. Okay, maybe I shouldn't call it phase 1 since the regulatories are here. I'm using phase 1 very loosely. They are more investigator-initiated trials for us to explore and understand how the therapy works, what are the manufacturing conditions, so on and so forth. So in a nutshell, it's really contrasting to what Natasha pointed out. Instead of reconstituting cells, we are trying to destroy certain cells that we want. We're trying to target things that we do not want in the patient itself. And instead of having an allogenic product where you can be applied to many, many people, our approach is extremely personalized. It's really custom made for every patient. And I think another aspect that really differentiates our approach is the fact that we are targeting hepatitis B as a tumor antigen. So it's not like what the typical CARs that you see, where they will look for alpha-fetoprotein, glypican-3, or other of these overexpressed fetal markers as a target. We are looking for a viral antigen that is, through the natural life cycle of the virus itself, gets expressed on a cell. The cell starts to be flagged up with all these antigens, and it could be recognized by our TCR therapy. So that's really kind of our approach in a nutshell.
Great, thank you. No, that's very exciting to see these two quite different approaches, for I guess different diseases as well. But I think some of the key differences, we heard about just now were, of course, the concept of allogeneic or essentially off-the-shelf therapies. The same treatment can be applied to all the patients versus a very personalized approach that Anthony's team is working on, which is taking the patient's own cells as the starting raw material to make the product. So obviously, very different targets, very different cell production source. So maybe Lily, I can bring you in now to, from a regulatory perspective. I know you said that you guys have some new guidelines, and it's very exciting. But in terms of, how does regulatory treat these two product types? Do they fall under the same guidelines, or are they, differences in certain characteristics you assess them on?
Okay, thank you. Yes, so this area is a very exciting field. So maybe I'll just give a little bit back in the history, like the first successful treatment of leukemia using bone marrow transplant actually started in 1959. And yes, way from there, and then now it's actually become a standard treatment you're giving for for leukemia.
Yep.
So, and yes, so and then a few years later in 1996 they started the first use of cord blood transplant in adult for leukemia as well. And as Anthony also has mentioned about anti-CD19 CAR, so this was create quite a big hype, like just slightly more than 10 years ago, whereby from clinical studies that shows many promising results using such a chimeric antigen receptor T-cell products. And from clinical studies, and up to now, currently there are 4 approved anti-CD19 products approved, and 2 anti-BCMA CAR-T products approved in US or Europe. So it's a pretty exciting results, just a very short span of like just more than 10 years. Yes, So with such many promising clinical outcomes for such novel therapies, but there are also, we hear also many cases from the media that there are many people using unproven stem cell therapy into the patient. So hence major overseas jurisdictions actually start to implement their regulatory framework for cells and gene therapy to have a certain oversight to such products. So in US, they start to implement the regulation of human cell, tissue and cellular tissue-based products regulation started in 2005. Yeah, so in Europe, it's like 3 years later to implement what they call the advanced therapy medicinal product regulation. Yeah, and then they divide the products into 3 categories: somatic cell therapy medicinal products, gene therapy medicinal products, and tissue engineered products. Yeah. So in Australia, they also have their regulatory framework. They call it biologicals. That means anything that has human cells or tissues is under this framework. So this piece of regulation is started in effect in May 2011. So they also categorize their products into four categories. So the lower risk will be in class 1, and then the higher risk will be in class 4. Yeah. So back around the clock, the globe. So now is HSA. So we implement our regulation last March 2021. So we also classify our products similar to the overseas jurisdiction based on 3 categories. So based on the degree of manipulation. So whether your manipulation is just basic cell separation, or is it going to be a more complex, like gene modification? And then the second category will be based on the intended use. So intended use means what is it going to use for? So one example is if I'm using like a bone from a cadaver donor, you just basically just sterilize it, and then you're going to maybe make it into a paste or make it into a chip and then formulate into a product and for orthopedic purpose. So bone and is for orthopedic purpose, then this is called homologous use. So this is, we will think this is a lower risk product. Yeah. So for the higher risk product, okay, the higher risk will be like if you are using bone marrow cells, but using for cardiac repair, like you collect bone marrow cells and you propagate them, and then you try to harvest the cells and inject into the heart. So we will call it is non-homologous use, because the original intent of the bone marrow is not meant for cardiac repair. Yeah, so this is the second category on intended use. So the third category is whether the product is combined with a medical device. Okay, the medical device in this context, it could be a scaffold. So it could be a polymer. It's a 3-dimensional polymer whereby you can seed the cells into the scaffold and then grow them up. And then you will implant the whole construct into the patient. So the scaffold itself is a medical device. So if you are combining cells with a scaffold, that is a combination product. Or if you are combining cells with a therapeutic product or a drug, that is also a combined product. So basically, we classify the products based on these 3 criteria. And then we have, so we have 2 classes of products. We have the low risk, which is called class 1, CGTP, we call it in short, and class 2, which is medium and higher risk. Yes, so we titrate, we have one piece of regulation, but we titrate our regulatory controls based on the product class. So we will have less control oversight for the lower risk products, but we have more for higher risk products. Yes, so the intent, overall intent for this regulatory framework is to encourage the product developer to bring these novel therapies to the market, but at the same time, we still have regulatory oversights. And that's why we take a risk-based approach, depends on how risky is the product. Yes, and of course, this framework has to be suit for this unique characteristic of the product characteristic, because like what Anthony is saying that one batch can be for one patient. I mean, certainly for such kind of products from Lion TCR, yeah, or for allogeneic. So in this case, there will be some differences. So that's why when we review the product, we will look into these criteria. Yes. So and then currently for manufacturing of actually the higher risk products is still quite manual. Actually, the processing is quite manual, and then so that's why you need to have a process you have control in place. You need to have in-process testing and to ensure that each batch of product is actually consistent, and especially not to forget because your starting materials is actually from the donor. So at the very starting, you already have donor variability. And then that's why you may have a lot of variation during the manufacturing. So it's really important to have proper product characterization from the beginning, and then to ensure you will have a proper process validation to ensure there's minimized batch to batch to ensure there's batch to batch consistency. Yes. So another point maybe I can talk about is because there's on the gene therapy products, some of them they're using lentivirus vector. So imagine the piece of transgene will actually integrate into the human genome. So that means your product is actually long-term persist in your body. So there is a need to have long-term follow-ups for this patient. So it can be as long as up to 15 years. Yes, and in our regulation, we also emphasize on traceability. Yeah, to ensure there is a traceability of records for more than 30 years after the expiry of the products, right from starting from where the sourcing of the materials, the procurement, the processing, packaging, and where the product is going to lead to who is the patient. Yeah, so the whole controls have to be proper traceability in place.
Yes.
Yes. So
yeah, thank you. That was very, very informative actually to hear about how regulation classifies these different products based on certain risks and degree of manipulation and process required. I think Anthony has a question. Maybe you can.
Yeah, so I mean, since we are diving into the regulatory perspective of things, so I mean, what struck me when you said was that you categorize things into a lower risk and a higher risk product, and depending on the risk, you have a different degree of control. But to me, I think the statement "a different degree of control" is extremely vague.
Yeah.
So when you talk about high risk, you talk about gene therapy. You talk about cells that are persisting using lentiviral vectors. So now, for example, what we do in Lion TCR, we use mRNA electroporation.
Yep.
First question, is it really a gene therapy if it's only transiently there? Because the mRNA is labile, it gets degraded, and the cell returns back onto its autologous form.
Mm-hmm.
And there is no combination of any drugs, there's no combination of matrix. You are taking it out, manipulating it outside, putting back in.
Yes.
So that's one dimension of,
correct.
let's say, complication, where there's no lentiviral.
Yes.
The cells, once it goes inside, well, it persists, but it persists as like normal cells.
Yes.
In this aspect, what does at least HSA view this as? Would you consider this as low risk? And to be honest, my personal opinion is that it's extremely low risk because it's transient, it's your own cells, it's not allogenic, there's no other things inside.
Yes.
And that's why my question is, when you say less control, what does the less control really mean?
Sure.
And where is that line?
Sure. Yes, that's a very good question. Maybe I can clarify further. So yes, that's the challenge when you mention about whether mRNA electroporation, so the end product, do you consider it a gene therapy product or is it a cell therapy products? So actually, in the different jurisdictions, there's just different way of classification. So that's why for us, that's why we don't have this category, whether is it unlike like EU, they have somatic cell therapy, gene therapy, tissue engineering. What we have is just low risk, class 1 CGTP and class 2 CGTP. So the low risk I'm referring to will be the bone graft for orthopedic purpose. So it, like I mentioned earlier, from cadaveric donor, you have the bone, you just sterilize the bone, crush it into chips, and then use it for orthopedic purpose. Or it can be skin from also cadaveric donor when you just sterilize it. So there's minimal manipulation, no manipulation, basically just sterilize it, package into final product, and then use it for burn indication. So we take that as low risk. So in this category of products, there's no need for product registration. We just need you to submit a product notification to us, yes, with some supporting documents. And there's no requirement for manufacturer's license. There's no need for importers and wholesaler license. You just need to submit a dealer notice. Yes. So what product, anything that is more than minimal manipulation, and it is for non-homologous use, it will be straight away classify it as class 2 CGTP. So that everything will kick in. So it consists of your lentiviral based gene therapy products, and also what Lion TCR is doing, and also iPSC based product as what Nasha is working on. So these are all class 2 CGTP, but you will need to do, you need for product registration, you need to come in for product registration in order to supply it into the local market. Or if you want to conduct a clinical trials, you need to come to us to get authorization. Yes, I hope this clarifies.
Yeah, sure. But that also automatically means that most of the adoptive cellular therapy products are immediately classified under high risk or
Yes.
beyond low risk. So
Yes.
then my follow-up question is, does HSA see a need to stratify what these high risk products are? So because, like, again, if I can understand that if it's allogenic, if it's lentiviral, if it's not the same, you mix it with other things, it's considered high risk. Of course, from your perspective, it's fine. But then, is it reasonable to then say that all cellular therapy products, in this case, are immediately above low risk? Because the low risk examples that you put there are essentially really very, very minimal manipulated products, which is really like bone graft, skin graft,
Yes.
so on and so forth. But anything that is, to put it very layman, any advanced cellular therapy products is almost immediately classified as beyond low risk. What Natasha is doing, what I'm doing, so on and so forth.
Yes.
But then my question to HSA is, do you then see a need to further stratify this framework to try and accommodate more efforts into this space, considering that you say that the existence of the framework is really to encourage further development and further progress in the field itself? When, in my opinion, if this dichotomous separation of low and beyond low seems more to stifle any progress than to actually promote progress.
Actually, we thought about it.
I'm so sorry. I'm being quite difficult here.
No worries about it, because I mean, this question actually come to us also when we way before we implement the regulation. We actually have several public consultation with the clinicians, with the product developer as well, and to discuss on how we stratify our products and the rationale. So yes, so the challenge, one thing is we try to make it a simple framework. That's why we have only two buckets, a low and a high, because we feel that the more you stratify, you create actually more gray products, whether you be fall in here or there. So we just want to make it a simple product, a simple way of categorization. But of course, in terms of long-term follow-ups, okay, let's say for that's why I've said long-term follow-up for gene therapy product, where if you're dealing with lentivirus vector, then you may need to have 15 years of follow-up. But if you're just dealing with cell therapy products, we should know that the cells will not persist too long, very long in your body. So perhaps the follow-up will be shortened. So these are the different that we will titrate accordingly to the type of products. because we can anticipate cell and gene therapy products is really a vast bucket. Yeah, so it's really a wide range of products. So it really depends on the product, and we have to put in the controls accordingly. Yes.
I mean, perhaps I will throw the discussion down to Natasha, because her approach instead, she would prefer the cells to persist forever and ever in her approach. So then what is the take of HSA then? Because then it's a complete different direction, right? Since now in her case, the longer it is, the better. But does it also mean that they have to follow up forever and ever and ever?
Sorry, maybe I'll just interject. So it's really case by case basis on how I mean, definitely for gene therapy products, you have to follow up for 15 years. But for iPSC, it really depends on your products and whether you have study to support how long it actually persists, perhaps from studies from your non-clinical, your animal models, or maybe from, if you have, to support that, what you claim.
Yes.
So,
I would be happy to chime in here. I mean, I was laughing quite a lot because it looked like it was a startup turning to a consultation session with HSA, which I think a lot of people like myself and Anthony are very interested in, to have this deeper conversation with the regulatory body. I think what I mean, no doubt in terms of the therapy that I'm developing, it's a high-risk product. There is substantial cell manipulation involved. I think the challenge that for us at the early stage that we face in terms of iPSC-based cell therapies is, for example, we are trying to generate iPSC cell bank, right? A cell bank that is suitable for use for further manufacturing to derive the cell types of interest. And I think in an ideal scenario, of course, the cell bank will also be generated in a fully GMP compliant manner, because then you have, all the documentation, all the processes done right in I guess the safest, highest standards. But in this case, it is very challenging for us because to generate iPSCs, you already require a lot of resources, and it's a huge effort, and yet the iPSCs are not the cell type that you are transplanting into a patient's body. The iPSCs will be, again, substantially manipulated into whatever cell type of interest the company is developing, in our case, beta cells. So the beta cell product is the one that you would, do all your QCs and all your, the testings that are required for the safety aspect. But yet, I think that it is unclear to us how much compliance we need to have at the iPSC stage. And I think I generally think Singapore, of course, tends to be more conservative, and of course the higher standard you can go for, the better. But this does require a lot of resources already, given that clinical grade or GMP grade iPSCs are not even readily available. So that's one challenge we face in terms of trying to evaluate what the regulatory process that we'll need to go through is. And the other thing I would like to mention is that for us to generate beta cells from the iPSCs, there is a very long process that requires already a lot of ancillary materials. I mean, we're looking at 20 to 30 small molecule compounds, and for each of them to need to be acquired from a GMP compliant source facility in the first place, sometimes that's not possible, because a lot of these compounds are only manufactured for research use, and I think the vendors are not keeping pace necessarily with synthesizing these molecules in a GMP compliant manner, just because the market size is not huge at the moment. So I think we're going to face a lot of challenges with each and every ancillary material that we use as well. So I think so in a nutshell, what I want to say is that, we are, as a startup, very thankful that HSA and other partners came out with this regulatory guideline and framework for cell and gene therapy products. I think that only speaks to the fact that we are at a very timely, a very good time to enter this space and to work together with regulatories and other regulators and other industry partners. But I hope that it would be a continuous conversation between the vendors, the regulation, and the startup, the technology developers, to try and find the best way forward, but also the most efficient way forward to materialize these cell therapies. And I guess the last point that I want to mention, because it was mentioned in terms of the follow-up, the length of the follow-up that's required. Of course, as Anthony alluded to, yes, we want our cells to last in the body as long as possible, because this is regenerative medicine. But it's not possible for us to do preclinical studies in animal models for years and years to show that they are safe before you start any human clinical trials, because then the progress is going to be very slow and people are just waiting. So I think there is already progress in the field. I mean, the fact that there are 20 to 40 iPSC-based clinical trials internationally shows that there is progress being made. Again, it will come down to different stakeholders recognizing the challenges and trying to help us to move forward in the most efficient way possible.
Yeah, thanks, Natasha, for summarizing that very nicely. I think, obviously this is an evolving space, and, as new therapies come up and different scenarios are presented, I'm sure the regulatory authorities will be responding in due course. So maybe we could, focus the attention now on some of the challenges that, you guys have faced in this space. One of the key areas has over time come up to be manufacturing. So obviously these cells are, very different therapies. How do you set up the manufacturing process? As Lily alluded to, manufacturing and process development is something they also look at. So at what stage do you start to, finalize, fine-tune your manufacturing processes for these cell types, and what are the challenges? Anthony or Natasha?
Sure, I can go first. So, like you said, at what stage do we start to really nail it down and start a let's call it a fixed process there? So, I think what is slightly bit different from a TCR therapy perspective than compared to general, let's call it the CAR T-cell therapy. So, similar approach, slightly different technologies. Well, the real difference is the fact that the CAR T is an antibody. It recognizes one particular protein. If I were to change the CAR T, I can make another antibody that recognizes the same protein, or I can make another antibody that recognizes a different protein. Now, when it comes to T-cell receptors, what happens is that one single protein can generate many different epitopes. And these epitopes are presented in conjunction with different HLA molecules. So in essence, you can have multiple TCRs or T-cell receptors that recognize the same protein. So from the scientific perspective, well, if they are recognizing the same protein with the same, it's just a different epitope, basically a different part of the protein, still the same protein, just a different, maybe in the front, in the back, or something like that, with the same HLA, well, it's often considered as a separate product. So immediately you can see the massive challenges involved in product development, manufacturing, even though the process is exactly the same. So from your question, the locking in the process is not so big a problem for us, because the process of growing the T cells, electroporating it, when to characterize it, it's pretty much fixed. We are just changing the TCR. We are changing the instead of putting an mRNA for this TCR, you put an mRNA for another TCR. That's all. But the fact that in multiple jurisdictions, not just in Singapore, it's always considered a separate product, and therefore you require another round of all this characterization, makes it extremely difficult for us to ask very, I would say, important question. What should we target, considering that you have this protein, and we already know from all the basic science that not the entire protein is equally expressed as an epitope for the T cells. Some are better than others. So if we do have in our library multiple T cells targeting different parts of the protein, it's extremely difficult even for us to just say, where should we target from the library that we have? Because I'm sure you know, characterizing one is an extremely huge amount of effort to do one. Now, just to answer that question, it's like as though we are developing 3 different products, 4 different products, and it gets worse and worse. So then we even toyed with the idea, what if we make all the TCRs, mix them together so we have a single product where you have T cells, 4 different types of T cells with 4 different T cell receptors recognizing the same protein. Can we do that? Or does it make it even more complicated to assess? Now you have things recognizing 4 different things inside one cocktail of stuff. It gets even more complicated. And that's why from that, locking in a manufacturing process for us, not a problem. But when it starts to really go down into the science and say, what should we target? Is it more effective to target this compared to another one? That's the big problem that we're facing. So a lot of the trials that we do, we essentially have only one target, one single epitope.
Yeah.
Then because of the HLA and everything, it makes it difficult to recruit individuals. It makes it very slow. It makes it very, to do a trial where we treated, I don't know, 8 patients, it took us 2-3 years to complete.
So I guess in your situation, the process may be more straightforward, but I think the product development and the trials, the clinical trial aspect is really the challenge. Maybe we can just move to Natasha in terms of what are the challenges she faces in terms of either product development or manufacturing for your technology.
Yeah, so I mean, I think this is it's interesting that we have different perspectives coming for different types of cell therapy products too, because in our case, so as an early stage company, when we're trying to develop these iPSC based products, so our case beta cell products, my main focus of course is cell functionality and safety, because I think these are 2 important things that you have to address no matter what if you want to progress as a technology and a company. So that's where a lot of our efforts are spent, trying to get your proof of concept data in vitro as well as in vivo. And in fact, in vivo, we are working constantly with mouse models, but as the field progresses, we already hear feedback that mouse models are not enough, because apparently mice are very easy to cure. So I think the standards are just becoming higher over time, as well as well, definitely more challenging, because the standards are almost for large animal models, where in our case would be non-human primates. And so we are driven to work with some collaborative partners to do our POC studies in large animal models as well, which, I mean, we fully understand the value of doing that. But what I want to say in terms of the challenge that comes about is, as we're doing fundraising, so because that's an important aspect for any startup, we speak to more stakeholders, and a lot of questions that we get actually relate to the manufacturing scale up as well. Because coming back to the point of tackling chronic diseases, no doubt it's a huge market size. I mean, for the diabetes market, it's a $1 billion market size, even if you're the 2nd or 3rd company in the market. So the questions we get a lot is, can your process be scaled up easily? And I think that's not an easy question to answer for a cell therapy product, as opposed to perhaps other biologics, drugs or antibodies, because cells, which are, I mean, we define here as living drugs, I mean, they are very sensitive to whatever activators or inhibitors that you put on them, and there is some inherent variability in how cells behave. So while we try to streamline and make our process as consistent as possible, doing it at a small scale right now and proving great cell functionality and safety, once we try and scale it up to say culture flasks, bioreactors, I think they might behave very differently. So I think that's a difficult situation that we're at, that we have to think about now. So, do we have to try our best to develop the protocols in a scaled up format already and show that that works? But in that instance, it is already very expensive and resource intensive. So I think that's one of the challenges that we face. Then, yeah, that's scale up. So I think the other thing, obvious one that I have to say for iPSC based products would be the safety aspect, because iPSCs are up there in terms of the questions on that we get on safety, because they have inherent tumorigenic potential. I think, as I mentioned just now, there are already 20 to 40 iPSC-based interventional clinical trials globally. And I think that is very promising, because that shows that, the labs, I mean, the teams have done much work on establishing the safety of their cell product to be able to run these clinical trials, and of course, continuous follow-up. So for us, I mean, even though we have competitors, but I think we are very much hoping our competitors to succeed as well, because if they don't, that would be a huge hit on the trust in iPSC-based products. So I think we're watching this space very closely, as with the investors as well. I think a lot of investors that we speak to are very, have an arm's length from iPSC-based products until there are some very promising clinical trial results that come out. And I mean, for those who are not very familiar with this area, that are already phase 3 trials. I think Japan is one of the leaders in some of the iPSC-based products in clinical trials. So I think we're all watching this space closely. And yeah, I guess that's what I can share for now.
Yeah, I mean, I think that's very good, nice insight into, when do you switch your process, and when do you scale up from the very beginning, and then do all the trials. So maybe Lily, you can jump in from a regulatory perspective. I'm guessing for any clinical trial, and then for the clinical trial approval, but then registration approval, you would require the same process the whole way through, or how does regulation sort of look at it?
Okay. So of course, locally, if you want to submit a first-in-man clinical trials, that is for class 2 CGTP, high-risk products. So you need to submit a clinical trial authorization application to us, and you need to compile your trial protocols, your investigator brochures, another important documents, which is the CMC dossier, which is actually the quality dossier, which actually compile of what, starting from donor screening testing, manufacturing process, what are the in-process testing, what are the release tests that you're going to carry out on the products, what about the shipping, how the product formulation, and then your storage condition, and whether you have stability data, and how you prepare the product at the bedside. So this is a big quality dossier, which actually during the past 10 years that we evaluate clinical trial application, we found that some local investigator may have some challenges in compiling these documents. Yes. So we actually have published a guidance documents on how you can compile this piece of document. Yes, we give what you're supposed to incorporate in each section. So at least give some clarity to the applicants what documents that we require. Because in the past beginning, we may end up have too many rounds of queries, just yes, because they may not be clear understanding what documents that we require. So that's why we need to we publish this guidance documents. Yep. And of course, for the my colleagues in the audits and licensing branch, they also has published guidance documents on the manufacturing, good manufacturing practice for cell tissue and gene therapy product, taking into consideration of such complex products, and there's a lot of variability. And actually, they also has adopted a risk-based approach, whereby you can take a look at the document, and there's a table 1 to show actually what are the GMP requirements for an investigational cell CGTP product versus a registered product. So there is a titrate differences, the expectation. Yeah, yes. So that is something you can take a look as a reference. And if yes, so another thing that maybe I would like to mention is that we welcome product developer or any investigator who want to submit a clinical trial application. They can actually request for a pre-submission meeting, or maybe even earlier, even way during their product development, they can actually request for a meeting for scientific advice or regulatory advice, like they can show us what is the non-clinical package that they have, and then maybe that is a more one-to-one, because that will be based on the product. So you give us some background of the product, we can advise accordingly, like how long you should monitor your products, how long is the patient follow-up, the duration, and what are the expectations. So that will be easier, rather than it will be tailored really to your product, because every product is different. Yes.
Exactly. I mean, I think that's the really takeaway from today, I guess, is that every product is quite different. Yes. And, you know, in terms of its biology and therefore subsequent manufacturing processes, product development stages, and regulation, of course. But, you know, it's great to hear that you guys are starting to put together some guidelines around, you know, what documentation needs to be done. So hopefully, you know, all the innovators with different therapies can, you know, be able to chat with you and further that dialogue. So maybe, you know, coming to the close of the hour session, we've just scratched the surface really of some of the challenges, but maybe as a wrap up, maybe each one of you can just say one or two sentences on what you think is the next 5 to 10 years, and what do you hope they will bring for this field, and what do you hope to see?
Okay, I can go. Yeah, I can go first. So I think I speak to everyone who works on iPSC development, and I think for sure we all expect that there will be more clinical trials based on iPSC derived cell types, and I think the potential for using iPSCs for regenerative medicine is huge, and I think a lot of people do believe in that. And so we hope to see more success cases, and I think we are already starting to see some of them in some specific cell types that are, I guess, more straightforward to derive and to quality control as well. So I think that will be a boost to the whole sort of industry for iPSCs. And what I would also like to end off saying is that I hope that if there are investors in the audience, or clinicians in the audience, or other stakeholders such as the vendors and the material suppliers, to kind of help us and work together with us to try and develop these very complex cell therapy product, and also be mindful that it is very challenging and very complicated to make these products work, and to, take the risk and help us push the boundaries. I know in Singapore, the ecosystem is smaller than in the big players like the US or the technology intensive countries like Japan, but I hope that in Singapore more and more people would be more open to supporting such therapeutic products, so that we can make progress.
So
we have a few minutes left. So,
I think from me, clearly there's plenty of trials for CAR T cells, TCR therapy, with this idea to destroy things in the body itself. So what I think people can see is that it works very well for liquid tumors, but for solid tumors, it's still a huge challenge. There's still many questions there. People are still asking, have we reached a plateau? Is it really possible to destroy in terms of solid tumors? And should we really explore combinatorial therapies and things like that? So clearly you can see that the trend is getting more and more complicated. So clearly things like regulatory and everything would have to keep up with this kind of complexity, and really see how they can fit in to try and really expedite or promote the investigations and the studies into it. And clearly for investors' perspective, like what Tasha said, that yes, the stakeholders and the local community is definitely way more conservative, and there's definitely a lot of resources that needs to be implemented to do. As you can see, it's extremely complicated, it's extremely difficult to do. So hopefully we have plenty of funding, easy regulatory, and successful science.
The dream.
Yes.
And
Maybe I just add something. Yeah, it's true as what Anthony say. Yes, actually, regulation is always lag behind the innovation. Yeah, it's always a few steps behind. Yes, we are trying very hard. So I think currently we see maybe there's more more innovative therapies such as gene editing products that may be coming in. So yes, as a regulators, we need to keep abreast to all the product developments, and also we encourage interaction with the local product developer as well. And we also have actively communication with our other overseas regulators in a different platform where we discuss what are the emerging scientific developments, share the best practice. So this is what as a regulators we are actively working on. Yes.
That's great. Well, this brings us to the end of the session today. Thank you very much to all the speakers for their panelists, and also to all the listeners. And with that, I'll just pass it back to Brian.
Okay, thank you so much, Mayura. Thank you as well to all our speakers today for the insightful and knowledgeable discussion. I think I speak on SGInnovate's behalf to say that it was a pleasure to have all of you here today to share your topics, to share insights on such an important topic. For our audience today, do take note that we'll be sending out a link to the recording to this session in our bi-weekly newsletter, so do keep an eye out for that. And with that, we've come to the end of our webinar today. Thank you all for spending your Thursday afternoon with us. And on behalf of SGInnovate, I would like to wish you all a great week ahead, and we look forward to seeing you again at our next event. Bye, everyone. Thank you so much, everybody.
Thank you.
Bye-bye.
Thank you very much.
Thank
Bye.
you.
Bye.